Welcome to Polyhipster. In our last video, we talked about G protein coupled receptors. Here, today, we will be discussing various other receptors in brief. So, let's starting with the tyrosine kinase receptors, which includes insulin and varieties of growth factors. Example IGF-7, EGF, NGF, PDGF, and FGF. These are cysteine-rich receptors and they play an important role in growth and differentiation of cells as well as intermediary. So, if we look at the mechanism of action here, once the hormone is recognized by the receptor, it causes binding and formation of hormone receptor complex, which leads to an autophosphorylation reaction, inducing interactions with intracellular adapter proteins such as SHC and insulin receptor substrates 1 to 4. HC is a signaling molecule that connects cell surface receptors to intracellular signaling pathways. Now, this causes activation of multiple kinases. An example here is RAF-RAS MAPK system, which is seen in insulin and AKT protein kinase beta pathway. These are signaling pathways which are a series of protein that regulate cell growth, differentiation, and survival. Next, let's talk about cytokine receptor family, which includes the growth hormone and prolactin receptor. Mechanism of action is simple. First, there is ligand binding leading to receptor interaction with the intracellular kinases. Example here is the Janus kinases, which phosphorylates STAT and other signal pathway. STAT being signal transduction and activation of transcription family, and other pathways like RAS, P13K, and MAPK. Once it is activated, it will translocate to the nucleus and it will stimulate expression of certain targets. Now, next we are going to talk about serine kinase receptors. These serine kinase receptors mediates actions of activins transforming growth factor beta, Mullerian inhibiting substance that is anti-Mullerian hormone and bone morphogenic proteins. Here, the receptors 1 and 2 signal through the protein's SMARTs, which has two roles. That is, first being transducing the receptor signal, and second is acting as transcription. They act primarily in a local manner, that is, in a paracrine or endocrine manner. Example, follistatin. The function of follistatin is to inactivate growth factors and restrict their distribution. The function here is opposing to what we learned in the other receptors before. Finally, let's talk about the nuclear receptors. All nuclear receptors ultimately act either increase or decrease gene transcription. The location of nuclear receptors are at two sites, first being the nucleus, second in the cytoplasm. The cytoplasmic nuclear receptor, they translocate to the nucleus only after binding with the ligand. The structures of these nuclear receptors are studied using X-ray crystallography. Most of the nuclear receptor bind to DNA as dimers. Each monomer recognizes a DNA motif as a half site. Now, so there are two types of dimers, homodimers and heterodimers. Homodimers, they are mostly seen in steroid receptors such as glucocorticoid, estrogen, progesterone, and androgen receptors. Their half sites are palindromic in nature, whereas heterodimers are seen in thyroid, retinoid, peroxisome proliferator, activator, and vitamin D in combination with retinoid receptors. The receptor specificity of DNA sequences is determined by three factors. First is the sequence of half site, second, orientation of half sites, whether it is palindromic or direct repeat, and at last, the spacing between half sites. Carboxy terminal hormone binding mediates the transcriptional control. These receptors are capable of mediating dramatic changes in the level of gene activity. Now, The tissue specific responses caused by these agents in the breast, bone, and uterus appear to reflect distinct interactions with co-activators. Now, after interaction with the co-activator, the receptor co-activator complex stimulates gene transcription by several pathways, including first the recruitment of the enzymes. Example histone acetyl transferases that modify chromatin structure. Next, interactions with additional transcription factors on target gene. And at last, direct interactions with components of general transcription apparatus to enhance the rate of RNA polymerase II mediated transcription. That's all for now. See you soon with more lectures on general endocrinology.